biomolecules. Introduction. Living systems are made up of various complex biomolecules like carbohydrates, protein, nucleic acids, lipids, etc. Proteins and carbohydrates are essential constituents of our food. These biomolecules interact with each other and constitute the molecular logic of life processes. In addition, some simple molecules like vitamins and mineral salts also play an important role in the functions of organisms. Carbohydrates. Carbohydrates are the organic molecules that are composed of elements carbon, hydrogen, and oxygen. These carbohydrates are referred to as saccharides. Carbohydrates are classified into three groups based on the number of sugar units and upon their behavior towards hydrolysis. They are monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides. These are the simple carbohydrates that cannot be hydrolyzed to simpler compounds. They are sweet to taste, crystalline, and soluble in water. They are commonly known as sugars. They are further classified based on the functional group and number of carbon atoms. They are aldoses and ketoses. Glucose. Glucose occurs freely in nature as well as in the combined form. It is present in sweet fruits and honey. Ripe grapes also contain glucose in large amounts. If sucrose is boiled with dilute HCl or H2SO4 in alcoholic solution, glucose and fructose are obtained in equal amounts. From starch. Commercially, glucose is obtained by hydrolysis of starch by boiling it with dilute H2SO4 at 393 K under pressure. Structure of glucose. Glucose is an aldohexose and is known as dextrose. It is the monomer of many of the larger carbohydrates, namely starch, cellulose. It is probably the most abundant organic compound on earth. Structure of fructose. Fructose exists in two cyclic forms which are obtained by the addition of OH at C5 to the group. The ring thus formed is a five-membered ring and is named as furanose with analogy to the compound furan. Furan is a five-membered cyclic compound with one oxygen and four carbon atoms. Disaccharides. The two monosaccharides are joined together by an oxide linkage formed by the loss of a water molecule. Such a linkage between two monosaccharide units through oxygen atom is called glycosidic linkage. Sucrose. One of the common disaccharides is sucrose, which on hydrolysis gives equimolar mixture of D plus glucose and D minus fructose. These two monosaccharides are held together by a glycosidic linkage between C1 of alpha glucose and C2 of beta fructose. Maltose. Another disaccharide, maltose, is composed of two alpha D glucose units in which C1 of one glucose 1 is linked to C4 of another glucose unit 2. The free aldehyde group can be produced at C1 of second glucose in solution and it shows reducing properties, so it is a reducing sugar. Lactose. It is more commonly known as milk sugar since this disaccharide is found in milk. It is composed of beta-D-galactose and beta-D-galactose and beta-D-glucose. The linkage is between C1 of galactose, C4 of glucose. Hence, it is also a reducing sugar. Polysaccharides. Starch. They consist of repeat units of monosaccharides or their derivatives. These units are held by glycosidic bonds. These carbohydrates liberate large number of monosaccharide molecules on hydrolysis. Some examples of polysaccharides are starch, cellulose, glycogen, and dextrins. Starch. Starch is the main storage polysaccharides of plants. It is the most important dietary source for human beings. Starch consists of two polysaccharide components. They are amylose and amylopectin. Cellulose. Cellulose occurs exclusively in plants, and it is the most abundant organic substance in plant kingdom. It is a predominant constituent of cell wall of plant cells. Cellulose is a straight-chain polysaccharide composed only of beta-D-glucose units, which are joined by glycosidic linkage between C1 of one glucose unit and C4 of the next glucose unit. Proteins. Proteins are the most abundant biomolecules of the living system. Chief sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. They occur in every part of the body and form the fundamental basis of structure and functions of life. They are also required for growth and maintenance of body. All proteins are polymers of alpha amino acids. Amino acids. Structures of amino acids are the building blocks of proteins. There are 300 amino acids that occur in nature. Among these, only 20 are known as standard amino acids that commonly occur in proteins. Amino acids contain two functional groups. They are amino and carboxyl groups. The amino group is basic and the carboxyl group is acidic in nature. The carboxyl group and amino group are attached to the same carbon atom. Hence, amino acids are termed as amino acids. Classification of amino acids. The amino acids which can be synthesized in the body are known as non-essential amino acids. On the other hand, those which cannot be synthesized in the body and must be obtained through diet are known as essential amino acids. Structure of proteins. Proteins are the polymers of alpha amino acids, and they are connected to each other by peptide bond or peptide linkage. Chemically, a peptide linkage is an amide formed between COOH group and NH2 group. The reaction between two molecules of similar or different amino acids proceeds through the combination of the amino group of one molecule with the carboxyl group of the other. This results in the elimination of a water molecule and formation of a peptide bond CONH. The product of the reaction is called a dipeptide because it is made up of two amino acids. For example, when carboxyl group of glycine combines with the amino group of alanine, we get a dipeptide glycylalanine. Globular proteins. This structure results when the chains of polypeptides coil around to give a spherical shape. These are usually soluble in water. Insulin and albumins are the common examples of globular proteins. Primary structure of proteins. Proteins may have one or more polypeptide chains. Each polypeptide in a protein has amino acids linked with each other in a specific sequence, and it is this sequence of amino acids that is said to be the primary structure of that protein. Secondary structure of proteins. The secondary structure of protein refers to the shape in which a long polypeptide chain can exist. They are found to exist in two different types of structures, alpha helix and beta-pleated sheet structure. Alpha helix is one of the most common ways in which a polypeptide chain dorms all possible hydrogen bonds by twisting into a right-handed screw with that of an adjacent turn of the helix as shown in figure. Tertiary structure of proteins. The tertiary structure of proteins represents overall folding of the polypeptide chains, that is, further holding of the secondary structure. It gives rise to two major molecular shapes, namely fibrous and globular. The main forces which stabilize the 2 degrees and 3 degree structures of proteins are hydrogen bonds, disulfide linkages, van der Waals, and electrostatic forces of attraction. Primary, secondary, tertiary, and quaternary structures of hemoglobin are shown above. Enzymes. Life is possible due to the coordination of various chemical reactions in living organisms. An example is the digestion of food, absorption of appropriate molecules, and ultimately production of energy. This process involves a sequence of reactions, and all these reactions occur in the body under very mild conditions. This occurs with the help of certain biocatalysts called enzymes. The enzyme that catalyzes hydrolysis of maltose into glucose is named as maltase. Vitamins. Vitamins are the organic compounds required for the normal maintenance and health of an organism in minute quantities, and their absence cause specific deficiency diseases. These are required in diet in order to perform specific biological functions. There are about 15 vitamins essential for humans. Vitamins are designated by alphabets A, B, C, D, etc. Some of them are further named as subgroups. Example, B1, B2, B6, B12, etc. Vitamins are classed into 2 major groups based on their solubility. They are shown above. Some important vitamins, their sources and diseases caused by their deficiency are listed in the table. Nucleic acids. It has been observed that nucleus of a living cell is responsible for this transmission of inherent characters, also called heredity. The particles in nucleus of the cell responsible for heredity are called chromosomes, which are made up of proteins and another type of biomolecules called nucleic acids. These are mainly of 2 types: the deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Since nucleic acids are long-chain polymers of nucleotides, so they are also called polynucleotides. Chemical composition of nucleic acids. Complete hydrolysis of DNA or RNA yields a pentose, phosphoric acid, and nitrogen containing heterocyclic compounds. In DNA molecules, the sugar moiety is beta-D2 deoxyribose, whereas in RNA molecule it is beta-D-ribose. DNA contains 4 bases, namely adenine A, guanine G, cytosine C, and thymine T. RNA also contains 4 bases. The first 3 bases are same as in DNA, but the 4th one is uracil U. Structure of nucleic acids. In nucleosides, the sugar carbons are numbered as 1, 2, 3, etc. in order to distinguish these from the bases. When nucleoside is linked to phosphoric acid at 5 position of sugar moiety, we get a nucleotide. Formation of a dinucleotide. Nucleotides are joined together by phosphodiester linkage between 5 and 3 carbon atoms of the pentose sugar. The formation of a typical dinucleotide is shown in figure.